Welcome everyone to the current installment of our data dispatch series. Today we're very lucky to have two esteemed guests, again from University of Chicago and TTI. We have in our presence today speaking Dr. Mike Yu, who's a research assistant professor at TTI. Mike received his PhD at UC San Diego and also studied at MIT, where he has a degree in electrical engineering, computer science. Today, our second speaker, I should say, is Maren, who is an assistant professor in U Chicago Medicine. So we're going to hear about some interesting overlaps between machine learning and human health and disease. So today we're going to hear about the microbiome and applications of machine learning to understand how bacterial evolution plays a role in human health and disease. So I think I should turn it over to the experts now. Mike and Maren, please take it away, and thanks for joining us today.
Thanks so much, Nick. All right, I'll share my screen. Can everybody see my screen? I think that's an affirmative. Okay, so thanks again for that introduction. I'm Mike Yu, research assistant professor at TTIC, and I'll be presenting work that I've been doing with Maren, as well as Emily, a PhD student in Maren's group. And our work is on studying the role of rapid bacterial evolution in human health and disease. And specifically, I'll just upfront give a more specific title, which is about modeling and discovering plasmids. So as context for this work, what we're interested in is a growing appreciation in recent years that microbes inhabit and surround us everywhere. So on the left side, you see that many parts of our bodies, skin, mouth, the gut, hair, everywhere is inhabited by many taxonomic groups of bacteria. And on the right, this is a diagram showing the degree to which these microbes have been associated with many diseases, from obesity, cancer, neurodegenerative diseases. It's so many associations. It seems like with every year, the more people dig, the more disease associations they find. And a large driver of these studies is now by the development of these omics technologies that have allowed probing what are the components of the microbiomes at a deeper and wider scale. The flow of these studies is you take microbiome samples. These could be taken from human fecal samples, or they could be taken from the environment, from soil or ocean water. You pump these samples through a machine that sequences all of the DNA in these samples, and they produce what is referred to as the metagenome. This is the sum of all the genomes from the different microbes that compose the microbiome. The metagenomes, these data sets, have been growing very quickly over the years. Each of these data sets, each metagenome on a raw level is a few gigabytes in size, and so now there are thousands, tens of thousands of these data sets. They're reaching terabytes to petabytes level of data, and so there's a rich treasure trove of information. We can, if we analyze these data sets correctly, we can start to really dig into the microbiology and ecology of these systems. So in studying these microbiomes and making sense of them, what is often done is associating the components in the microbiome with some disease phenotype of interest. And so that's what I'm showing on the left side. You have some kind of feature matrix, say patients being the rows and columns being some taxonomic components like species inside of your microbiome. And you associate it to try to predict some phenotype of interest. Say this is inflammation or cancer or some other disease phenotype. And so that's been, many studies have been performing this sort of high-level approach. But one of the limitations of this way of thinking is that it only examines species, a specific kind of genetic unit in the microbiome. A more complete picture of the microbiome would examine other genetic elements, particularly mobile DNA, that is also important, functionally important in these microbiomes. And in this talk, we focus on a very important type of mobile DNA called plasmids that can transfer within and between cells. And so the end goal is that if we can identify and model plasmids, we'll have a more expanded feature matrix here of not only species or other taxonomic units, but we can include plasmids in this expanded feature set and potentially improve our predictions and understanding of phenotypes. So what are plasmids? Plasmids are a very versatile type of genetic system. They're often short and circular pieces of DNA that can replicate independently from the chromosome. The chromosome being typically what's thought as the main part of the genome. Plasmids, they're often transferred between cells, unlike the chromosomes, and they do this by this process known as often by this process called conjugation. In the top right, this is an image of two bacterial cells performing this process of conjugation, where a physical bridge, often a structure called a pilus, is formed between the two cells, and it is through this bridge by which the plasmid is transferred. In the bottom, we have a cartoon depiction of this transfer by conjugation, where the green circle represents a plasmid, and this is being transferred between the cells, whereas the purple circles are the chromosomes, and these sort of remain in their original cells. Plasmids, they're important for bacterial function because they can sort of augment the genome in many different ways. One of which is that the plasmids encode genes that provide antibiotic resistance. They might allow the bacteria to kill other bacteria or become pathogenic. They can also encode genes that provide metabolic functions. So plasmids are very important as they occur naturally in bacteria. And I'll just note that plasmids also form a bedrock of biological engineering. Often synthetic plasmids are constructed to deliver genes of interest. So we want to study the role of plasmids in how they shape the microbiome, how they shape the flow of genetic information across bacteria. But a prerequisite to studying this question is a much simpler question, which is, given some DNA that we sequence, just this random toy sequence I put here, did this come from a plasmid or a chromosome? And it turns out that actually this is not, there are crude approximations to this, but it becomes quite difficult to answer this very well. There's been many previous studies and data-driven models for understanding plasmids towards addressing this question. Some early studies were about categorizing plasmids into subtypes, like plasmid subtype A or plasmid subtype B. This isn't directly about the question of classifying plasmids from chromosomes, but nonetheless, these were important works for establishing components of plasmid biology. Some of this work consisted of generating these networks of plasmid similarity. So in these networks, you'd have a node represent a plasmid, and you have edges representing whether two plasmids share a high degree of sequence similarity. Other approaches did an approach called typing, where they would sort of identify subtypes based on canonical plasmid genes involved in the replication or mobilization of these genes. And as I mentioned, these are all important studies in understanding plasmid biology, but they weren't really directly about distinguishing plasmids from chromosomes. So it's not clear how well these studies would help for this task. Another sort of studies did address this direct task of distinguishing plasmids from chromosomes. A large degree of these studies, though, most of these studies, the way they did this classification is by decomposing the DNA sequence into these short windows of 3 to 7 nucleotides, what are referred to as kamers. So it's sort of taking your entire DNA sequence and blending it into really small pieces. Part of the problem, though, while this seems like a valid approach, this is a valid approach, but one of the limitations is that it doesn't directly, it sort of directly ignores some fundamental biology here, which is that the genome isn't just a blended soup of small kamers of 3 to 7 nucleotides, but they're actually modular, composed of these units called genes that provide specific functions. And I'll say across these studies, in my opinion, there's been some but very limited interpretation about the genetic architecture of plasmids beyond what is classically known about replication of the plasmids. There's limited interpretation about how other functions, as I mentioned, such as providing metabolic functions, as well as making the host bacteria pathogenic or other functions, how all of these are sort of tightly woven together. And so we sought out in this study to develop machine learning strategies, a new machine learning strategy to classify plasmids from chromosomes and along the way understand the architecture of plasmid biology. And our general strategy, sort of on a high level, one way to look at it is we model genomes, that being plasmids and chromosomes, as if they were human languages. So what do I mean by that? Here's a, let me take an aside and give an anecdotal task that's done in natural language processing, which is to rate movies. Well, to take movie ratings, the text, and infer what was the rating that was given. And so in this first 2 sentences, there's a good rating. The actor gave a spectacular performance. That maybe whatever that movie was, maybe is given a 5 stars. And then there's a bad movie rating where the review was the actor showed an abysmally flat range of emotions, and so that would be a 1-star rating. And so sort of on the simple way to understand what is going on is to identify these keywords of the emotion. That being here, spectacular sort of is the keyword indicating that this is a good movie, and abysmal is clearly that was a bad movie. And so on the first pass, that's what you might try to learn. This is how you might learn ratings. In the next example, these 2 sentences, it does. My point here is to show that this task can be a little more complicated because you can have what are known as words that have polysemy, which means they have multiple meanings. Here, the word complex occurs in both the good and bad movie rating. And so you can't really just use the word complex by itself. You have to look at the context of other words in the sentence. For example, masterfully or disarray, to distinguish whether this was a good or bad movie rating. So my point is here, though, is that relating this back to plasmids and chromosomes is that in trying to distinguish between these 2 types of DNA, on a first pass, sometimes it might help to look at sort of keywords like spectacular, abysmal. But in other cases, some words, some parts of the DNA are not 100% discriminative. You need to look at multiple words or the context in order to get a better understanding. So in the next slides, I'm going to talk about, along this train of thought, some preliminary steps we've taken to understand how genomes, how plasmids and chromosomes are like languages. And so I'll give 4 anecdotes on this. The first is to build a vocabulary of the genome in the same way we have a vocabulary of words like spectacular, abysmal. The next two steps are to examine the unigram and bigram frequencies. That is how how frequently individual words occur, as well as bigrams, how pairs of words occur together. And finally, we'll examine something called word embeddings, where you try to capture how words occur together in higher order contexts. So the first step, as I mentioned, is to build a vocabulary. And so this is conceptually very simple. You take your DNA sequence. This is, again, the toy example I showed on the left side. And what you do is you highlight substrings in this DNA sequence. Here, the example is the red, yellow, and green substrings. These represent genes, and genes being sort of the sort of these atomic units in the sequence that encode proteins and other molecules. And what we do is we take each of these genes and we define, sort of we label or annotate them by this ID. Here in this example, the red sequence is labeled COG1234, the yellow is PFAM8275, and green is de novo 2837. And what these labels mean is that they represent something called gene families. These are sets of genes that have a very high high sequence similarity. So all the sequences that are ever labeled COG1234, they are either exactly the same set of ATCG, the exact same sequence, or are very similar. Likewise for PFAM or this green labeled de novo. I'll come back to this and explain more of it, but I'll just quickly say that in the study, we worked with three kinds of gene families, specifically the red families, which are these what are known as COGs. There's over 4,000 of them. In yellow, we have this set of almost 12,000 families known as the PFAM families. And then in green, we have a set of over 400,000 what we'll call de novo families. They're called de novo is because we generate those ourselves. Okay, so now we've built this vocabulary. We've taken all these substrings and we've labeled them according to these green, red, and yellow sets of gene families. And the next step is to look at what is the frequency of these individual families. And so what I've plotted here, let's take the leftmost plot here. This on the x-axis is the frequency of each of these families, specifically the COG, the red families, as they occur in plasmids. And the y-axis is the frequency of these families in chromosomes. So most of these families, at least these COG, the red families, they occur on the left side. That means that they have a very low frequency among plasmids and a very high frequency among chromosomes, with an exception of just a very few 18 families. A very small fraction of families are sort of enriched in plasmids versus chromosomes. So if we were to try to use these families by themselves individually to sort of classify plasmids or chromosomes, there isn't sort of much working material here because most families are just chromosome specific and very few are plasmid specific. If we look now at the middle plot, this is just the same plot except we're looking at different set of gene families. It's no longer the COGs, those are the red families, but now we're looking at different sort of class, the green or PFAM families. Here it's sort of the same story where almost all of the families are hugging the left side, meaning they don't really occur in plasmids, but they occur a lot in chromosomes. So this is sort of a problem if we look at these two families, sets of families, COGs and PFAMs. On the right, and I'll explain more of I'll come back to this in later slides, but on the right, this is the same plot except we now use these what we're calling de novo gene families. These are gene families or vocabularies, words that we identify from scratch by sort of clustering all of the sequences, DNA sequences together. This sort of represents a more unbiased set of vocabulary of the genomes. And what you notice here is that now the points no longer hug on the left. There's some that are hugging on the left, but some they're hugging the bottom. And those on the bottom are the ones that there's more of these sort of represent potential markers of plasmids. So actually, I forgot to mention, maybe this was implied, but while these de novo families, I said we constructed them automatically by clustering all the sequences together. The PFAM and COGS are somewhat automatically generated also, but they were generated by other people in these reference databases. So they potentially suffer from biases based on what data they were generated on and what was sort of the model parameters there. Okay, now we can move one step up in complexity, go beyond unigrams or the frequencies of individual gene families, and look at bigrams, look at how pairs of gene families occur near each other. And what we see here again is sort of the ideal distribution, which is that we have some of these pairs of families. Every point here represents a group of a set of gene family pairs. We have some of these pairs on the left, those are specific to chromosomes, and we have some that are on the bottom, and those are specific to plasmids. So these again, so this is the ideal plot because we can use those on the bottom as sort of as markers. If we see them in a piece of DNA, then it's likely that that is coming from a plasmid. Oh, okay. So just as an example of what I mean by a bigram, this is an example bigram that is very plasmid specific. So we're looking at two gene families here, this TRA-E, transfer E, and transfer Y protein families. And this slide is showing where these two occur in two different plasmids. So in this Schumanella plasmid as well as this Ranella plasmid, I've highlighted in black where these two families occur. So here's the transfer Y, and here's transfer E in the first plasmid, and then there's the sorry, my Zoom bars are getting all over the place. The speaker bars. Hold on, I gotta move things. Okay, here we go. So the here's the transfer Y and transfer transfer Y and transfer E proteins in the second plasmid, and I drew these orange lines just to indicate that they're sort of this correspondence. They're next to each other in both plasmids, and I'll note that this isn't just the only two plasmids they occur together. They occur across many different plasmids, actually a wide diversity of plasmids across 4 different orders of plasmids. But what's interesting is that although they occur often in plasmids, you never see them together in chromosomes. It's not that they don't occur, like these individual families are used in chromosomes, but they're never together. They're often disconnected in different parts of the chromosome. So there's something interesting about their co-localization. And if I can examine more around this locus and see that actually there are these other genes, this transfer L or this type 4 conjugation system lipoprotein, transfer V protein, all of these other surrounding proteins also follow sort of a similar order, what's known as synteny across these different plasmids. So just to further dig into this example, so this was the first plasmid, the Schumanella plasmid. If I look at this locus, these genes that are all sort of conserved between those two example plasmids, I can further dig in, make this heat map, which is the genes by themselves, genes by genes in this locus, And what's plotted here is how often these two gene families co-occur. What is their bigram frequency across all the plasmids? And so what's being shown here is that all of the, many pairs within this locus have a high bigram frequency across the plasmid. So it's sort of a strong evidence that this set of genes here, this set of 7 genes, is actually sort of an atomic unit that is being transplanted from one plasmid to another. And just lastly, I'll just quickly show another example. These are, again, 2 plasmids in a specific biogram, this COG1175 ABC type transport, and this MM6 family. This is one of our de novo families that we constructed from scratch. And we can observe how they have sort of a similar ordering between these 2 plasmids, and not a perfect correspondence, but they're, if you look at the surrounding genes, they also are somewhat, the ordering is somewhat conserved. Okay, so now we've gained some intuition that there is sort of these systematic language patterns, unigrams as well as bigrams across plasmids and chromosomes. The last step to build further intuition is to create what are known as embeddings. This is a very important, popular form of modeling that's been used in natural language processing. And the concept of embeddings is well, embeddings is just a synonym for dimensionality reduction. So if you're more familiar with dimensionality reduction or principal component analysis, this is the same thing. It produces the same kind of thing, except it's a non-linear way of doing it. Principal component analysis and other kinds of, so older techniques like that are linear forms of dimensionality reduction. But what's the purpose, the optimization that's being done in these embeddings is you generate this vector or low dimensional embedding in Rn space to represent every gene family. So we'll say that the embedding is W sub I for gene family I. And we construct these embeddings such that the co-occurrence, how often, so the bigram frequency of these two gene families, that will be mostly, that will be captured or recapitulated by taking the dot product between the dot product of the embeddings of the 2 gene families. So so basically, if you another way of putting it, if you're not familiar with this idea, is that the sort of the geometric location of gene families in this sort of low dimensional space corresponds with how often they occur in these genomes. And so if we do this, our biological hypothesis is that gene families that are close together in this embedding space, that means that they occur co-occurring in the genomes, and therefore maybe they also have similar molecular functions. And I'll quickly draw, a lot of these ideas have been matured in natural language processing. Maybe this is an easier analogy to digest. So one of the signature examples for embeddings is finding this embedding where man and woman are these 2 points in the embedding space. And then you see king and queen, which is kind of like the man and woman version of royalty. They are equidistant from each other. They're sort of like you sort of almost just shifted the man and woman line to the right. And so the embedding space also preserves sort of these analogies or relations between words. And that is part of our goal, is to preserve as much of the contextual information in this space. So if we do this, this is, I cherry picked, this is the best example, and I'll show slightly worse examples, but I want to give the best example so you understand what's going on. That this, if we take a look at the gene families that are involved in the type 4 secretion system, which is a very important part in plasmid replication and transfer. So these are all the gene families that occur in the type 4 secretion system. If we look at where they occur in the embedding space in the top right, so here every point represents a gene family. the red points represents the type 4 secretion gene families. Almost all of the type 4 gene families are clustered around here in the top. So that's a good verification that this is this embedding is sort of recapitulating the important context, genome context. Now I can go beyond type 4 and look at other kinds of functions. Here I'm showing 26 different functional categories. For each category, I color all the gene families that are involved in that category. And so for some of these functional categories, for example, here at the bottom, this mobilome, prophages, and transposons, they're all kind of clustered in one part of the embedding space. That's what we want to observe. But in other cases, it doesn't work as well. So for some of these, like transcription or ribosome, it's kind of all over the place. So that, what this suggests is that there is important context to be learned, language modeling to be done, though there's still room for improvement. Okay, so I hope I've given you some intuition about how this is a very, this is like a language-like problem where we can use a lot of machine learning ideas that have been developed in natural language processing and other areas of bioinformatics that have approached similar questions to classify plasmids from chromosomes. And what I'm showing here is a diagram of some of the data formatting and analysis in our pipeline to do this task. And I'll go through some of these steps in the pipeline. I'll note that a large part of this pipeline is powered by Amvio, which is this very big software platform, very big and popular software platform for many types of microbial analyses and visualizations that Maren and Maren's group has been developing. So this has been a very good joint collaboration because we can benefit. I can benefit off of a lot of the expertise that Maren's group has put into the software and the intuition of microbiology. And we hope in the end, actually, that some of the modeling machine learning models that we are developing can be rolled back into this platform so that others can use it. Okay, so I'm going to go through some of the important steps in this data analysis pipeline, and I'll describe them. I'll describe these steps as solutions in a few different biases or challenges that we observed in this task, plasmid classification task. So there's, as we see it, 3 major challenges or biases. The first is that plasmids are typically shorter than chromosomes. So already, you're not really examining apples to apples. Like plasmids are just typically much shorter. And I'll dive in more details about each of these challenges. The second challenge is that plasmids, the genes are less represented in known gene family databases. This is actually something I showed already in a previous slide where I showed that if you recall, the COG and PFAM families were not really, they were mainly enriched in chromosomes. Those were those plots where I kept saying they were hugging on the left side, and there weren't very many gene families that were hugging on the bottom side, that were being enriched in plasmids. A third bias is that chromosomes in plasmids, they tend to be, if you look at reference databases, they tend to be overrepresented for very well-studied species. Okay, so how do we address some of these biases? The first bias, that plasmids are typically shorter. Let me show a plot to again explain really what I mean. This is a histogram of sequence lengths, where the blue histogram, blue bars represents the distribution of plasmid lengths, and the orange bars is the distribution of chromosome lengths. So you can immediately see that plasmids are typically much shorter, up to at most around 2 megabases. Actually, most of them are much shorter than that because this is a logarithmic plot on the y-axis. And chromosomes can span up to 12, 14 megabases. So this is a problem because you can perform very easy classification by just looking at the length. If I'm giving a short sequence, I don't need to know anything about the biology, but just say it's a plasmid. If I see a long sequence, I'll just say it's a chromosome. But that's true. There's the two problems with that. One, we have learned no biology, and second, when we go and sequence a bunch of DNA, we won't often reconstruct the entire genome, and so we won't really know what is the full length of the piece of DNA. We're just sort of examining a small substring. So to help address this bias, what we do is we break up the sequence into windows, into substrings of 50 kilobases long. And that's this part of this data analysis pipeline. And a second challenge is that plasmid genes, as I mentioned before, are less represented in known gene family databases. So what I'm showing here on these plots is very related to what I showed earlier. On this top left plot, this is a histogram showing how many of the genes among plasmids are annotated by COG families, which is one kind of gene family database, versus how many of the chromosome genes are annotated by these COGs. And so what this histogram is showing is that most chromosomal genes, so for every chromosome, about 80% of the genes in that chromosome, you can identify a vocabulary, like a word for that gene, 80% of the time, whereas plasmids is this wide distribution. For some plasmids, very few, if any, of the genes, you have no vocabulary for it. It's just some random substring. Now, if we look at the bottom plot, if we incorporate these de novo families that we construct from scratch, the picture is a little better, where now a lot of plasmids, a lot of the genes in plasmids do have a vocabulary. And this plot is just showing the same thing, so I'll skip that for the sake of time. Now, the third bias is that chromosomes in plasmids, they're very overrepresented for specific kinds of bacteria. This is actually a widespread problem in a lot of any kind of bioinformatics, that what is known in your training examples, if you're trying to set up a machine learning task, are typically just what is really well studied and was already known. So in this case, it's plasmids of chromosomes, often in, let's say, E. coli plasmids or chromosomes, or salmonella, or these are kinds of bacteria that are human pathogens, and so they've been very well studied. I guess there's more funding for that. But the problem is that there's all other kinds of forms of life that only have very few examples in these databases. So if we're not aware of this bias, we'll just be learning what is the genetic architecture of a human pathogenic bacteria. We won't learn about what are the beneficial bacteria, or maybe bacteria or plasmids that inhabit the environment. And so the way we address this is we group together, we cluster together plasmids of chromosomes that have a high sequence similarity, and define these sort of subtypes or subgroups, so that all the E. coli, more or less, plasmids or chromosomes are in one group, all the other plasmids are in another group. And so then we can assess, as I'll show later, we can assess how well we're doing across the groups, and not just by sort of the raw number of plasmids or chromosomes. Okay, so now I've gone through a lot of intuition, a lot of preliminary setup. Now we're going to get to actually some of the machine learning. And I realize it's already 40 minutes after the hour, so I'll speed this up. But I'm cutting to the meat of this, which is that we built some machine learning models to train plasmids versus chromosomes. And specifically, there's sort of two kinds of models we tested. This first set of models is where we generated a one-hot encoding. Sorry, let me take a step back. In any of this machine learning, you need to create features to represent your sequences. So in our first class of models, the features we used was what's known as a one-hot encoding of gene families. That's shown on the right here, where we say gene 1. In this example, gene 1 is represented by 10001, because the first bit, or the first location of this vector, represents whether gene 1 is present or absent in your sequence. Likewise, gene 2 has its own sort of one-hot encoding. Gene 3 has its other one-hot encoding. And what we can do is we can now, if we look at a sequence that consists of many genes, we can take what's known as a bag of words pooling, where we just simply add together all these vectors. So if we have a sequence that's gene 1, 2, and 3, that bag of words representation is just 1, 1, 1, 0. We can then take these features and pump it into some kind of model like random forest or an L2 regularized logistic regression to classify plasmids versus chromosomes. And I'll also say that we did try a different kind of feature space, not one hot encoding, but we used some of those embeddings that I showed before, these low dimensional reductions. And again, we either do a bag of words pooling, or we feed this into a deep learning architecture called the recurrent neural network, specifically a bidirectional LSTM. If you're not aware of those details, don't worry about it. The basic point of it is it's a little more complicated version of bag of words. It's not just summing together all your vectors. You're feeding it into this sequential model. If you're familiar with HMMs, it's sort of an HMM on steroids. And lastly, we compare it with one of the more recent models for this task called PlasClass. Okay, so how did we do? We tried a vanilla scenario where we took all our plasmids and chromosomes. 50% we made it the training data, 50% was the test or validation data. And here we're just using a very simple set of gene families, or very small vocabulary, which is the 4400 COG family. So we're not using PFAM or these de novo families. And what's being shown here is the ROC and precision recall curves for the performance. And what you see is that, well, first I'll note that the 4 of the models we tested, this OHE means one hot encoding, 8-dimensional embedding, all these different models, they all kind of hug the top left of this ROC plot. And the area on the curve is near 1. So that's getting to nearly perfect prediction. What was surprising is that one of these competitor models, PlasClass, did not do that well. And it's actually that the performance difference is better seen when you look at the precision recall curve. And so, you know, the 2 takeaways here is that, you know, we're already outperforming some of these previous models, but it looks like we're already topping off or saturating in model performance. We're getting almost close to near perfect prediction. And so what's happening? Is this problem actually very easy? Was it really trivial? And as I'll show, it's no. It only seemed trivial because of the way we split the data into training and testing. So what I just showed is if we take the plasmids and chromosomes, and they're color coded here, that the red plasmids are all very similar plasmids, and the blue is another group of plasmids, as well as the green chromosomes and purple chromosomes. We can split them. If you naively split them into training and test, what you might get is that you have basically the same plasmids in both the training and the test. And so you can get a very trivial model just by regurgitating or memorizing what you saw in the training data. A sort of better validation scenario is where we split the data by being aware of these biases, or this sort of internal clustering of our data points, where we just put the red plasmids on the left and very different blue plasmids on the right, and likewise for the chromosomes. Okay, so what happens if we do that? On the left side is what I showed earlier. This is the precision recall curve with what I'll call the naive split, which is pretty, you know, it's an easy task. Now, if we do a more, what I'll call the informed split, this is now a harder learning task. And now all our models start to get worse. So, okay, so now we made the problem harder. And what I'll say quickly is that we can actually now there's room for interesting modeling. So, because now we want to improve the performance again. And the way we do this is, I mentioned before, we were only using 4,400 families. It's a very limited vocabulary. If we now roll in a wider vocabulary, now over 430,000 families we can boost this precision recall curve. So it's not perfect anymore, but it's much better than before. And so, okay, so what I've shown is that we've built these models. We can learn plasmids in a sort of unbiased way. And lastly, this is the last slide. What we've been doing, is still ongoing, is taking this machine learning model and looking at metagenomes in the wild and examining trying to discover new plasmids. I'll just, on a high level, say we've done some of this, and we've started to characterize what we think are ideal plasmid candidates. So first, what is an ideal candidate? On a first level, we identified about 13,000 DNA sequences that contain a signature that looks like a plasmid. That's because our model said so. The models I showed earlier. We can further refine these candidates and look at which of them sort of occur abundantly across our metagenomes. We get about 4,000 of those. We can further wean this and examine which of these candidates are quite new. They don't look like anything in our known databases of plasmids. There's 2,000 of them. And then lastly, many plasmids are circular. They loop back on themselves. And so we can find that about 100 of them, there's evidence of a circular configuration. Okay, so the takeaways of this was that I hope you've learned that plasmids are an important component of microbiomes, and they have their own genetic architecture that we can we can classify and model as if they were like languages. And this is a very interesting and there's a lot of opportunities in this space because there's an increasing large database of reference genomes as well as metagenomes. And I'd like to thank Rob and Nick for organizing this, and the CDAC team for providing us with a seed grant to do this study. And I also want to thank my collaborators, Maren and his PhD student, Emily. This has been a very great collaboration. So with that, I'll take any questions. Thanks.
Thanks, Mike. That was great. Really enjoyed learning about the microbiome and connections to language processing. I never would have thought. So thanks for that very enlightening talk. I guess I can kick it off with a question.
Okay.
I was curious about the connections that you made between the genome and word embeddings and language embeddings, and that was sort of central to pretty much the whole approach. I was interested in sort of what naturally led you to that. I mean, what sort of makes a gene similar to a word, or a group of these? What makes it make sense to think about that as a bigram or an n-gram? And I guess
that's a great
question, language has a certain structure.
Yes.
And genes presumably have structure that's not necessarily the same as language, but apparently has qualities that are similar enough that mean that this kind of representation and these kind of embeddings can make sense.
Yeah, that's a really great question, and actually it's an opportunity for me to draw a deeper connection I didn't have time to touch on, which is the reason this is like a language is like in human languages. Human languages are modular. The semantics and syntax of human languages are modular and hierarchical. Language is not just a bunch of letters or phonemes or morphemes. There's a hierarchy to it, that you start composing letters together into words, and words also compose together into phrases or idioms or sentences to have higher order meanings. And likewise in genomes, you have very similar modular and hierarchical structure. Then on a very low level, raw level, you have nucleotides. Those are like your letters. And there's, of course, a lot of information just going letter by letter, but you do need to look at multiple scales simultaneously. So we go one scale up, letters in the same way, like letters in language form words. in genomes, you form genes because that's the way biology has evolved. That if the genome is sort of a memory tape, you don't just read one letter at a time. You read a group of letters to encode a protein or some molecule. And so there's sort of by evolution, there was structure imposed on a word or gene level. And looking at bigrams is sort of now, and embeddings is now kind of getting to phrases or idioms and higher order engrams or sentences, and understanding that structure. So likewise in biology, in these genomes, you have cellular pathways that have evolved that involve multiple words or genes together, because you can't just, you can't replicate an entire cell using one protein. You need multiple proteins working in concert.
That's super interesting. I never really occurred to me that the same kind of hierarchy that it exists in letters to words to sentences would exist in genes. That's fascinating.
Just perhaps add a little bit what Mike said. Another aspect of this that I find very fascinating. These genes come together and form pathways that can do certain things. For instance, nitrogen fixation, it requires a bunch of genes that are doing structural and catalytical things. So they, when they move around from one genome to another, they move around often together. So you find similar organizations of similar pathways, even in evolutionarily distant taxa. This is similar to saying you read different books from different authors, yet similar sentences appear to be in multiple books because they serve a purpose very clearly and very nicely. So people tend to use them all together and in the same form. So I think the connections Mike is drawing and then following up, there's a lot of benefit to include in our thinking of biological systems.
Cool. I had one other question, and I guess if there are others, and maybe Rob or some folks from the audience want to follow up too. But another was on your clustering of plasmids. I thought that was a pretty interesting way of sort of dealing with the very large number of those. And does that have a connection? Are there similar things that show up in language? Is there an analogy there?
Are you referring to this data split?
The earlier slide where you sort of circled the very high number. This one, yeah.
Oh, I see. This is related to the training test split. So I clustered plasmids and chromosomes together, and I use this grouping to then guide the splitting. So in training and test, so that the training test split wouldn't divide groups up. I
see. Okay.
But this I didn't mention it. The way I did this was actually quite an approximate and crude way of doing it. There's a great sort of a wider set of techniques for doing this in a more refined way, where you that tries to better learn that plasmids or chromosomes come in different taxonomic groups. Like you have plasmids that came from a specific species or genus or high order taxonomy. There are more sophisticated ways of doing that. I sort of took a very crude way just for the sake of dealing with this sort of the naive train test split.
Interesting. Cool. Thanks.
But I guess if maybe your question was, maybe I keep drawing these analogies to language. So I apologize if this is getting too much, but maybe the analogy would be kind of like topic classification, topic modeling in language, where you want to learn documents that are related to history or drama or science fiction or whatever, what have you. Your understanding of the usage of words and phrases and language may differ across these different topics. And so it's important to learn topic or taxonomic or group specific structures.
Cool, that makes sense. That's really interesting. Thanks. There's a question in the chat, which I think you might be able to see from Vanshika Gupta.
Okay. reading
Who
that.
asks if you could expand on how the system deals with differences between the gene sequences?
So I think there are different levels to that question. I'm not exactly sure what aspect, Vanshika, you're asking, but I think you might be referring to maybe genes. Some genes are very similar to each other. Like if it's a gene of 100 nucleotides, you might have different versions of this that are very similar, except they differ by 1 or 2 nucleotides. That was this concept of gene family that I kept alluding to, where we have this concept of a gene family which encompasses all the variations, slight variations of more or less the same or very similar sequence. It would be, I don't know if you come from a biology background, but let me just assume you come from an NLP background, and apologize again if this is too much, but if this were a speech recognition task, everybody says the word tomato or tomato or potato or potato differently. And so if you looked at the wave pattern, they would look slightly different. I say tomato, you say tomato, but we know that that's basically the same word. And so we would cluster them together. Here, we have variations of the same gene sequence or very similar gene sequence, and we approximate that this is the same, what we call gene family.
And it's basically encapsulated by gene clusters Mike generates by relying upon amino acid sequences. And the purpose of this entire thing, of course, is to be able to identify genes that are not identical to those we know about at the sequence space, yet they are similar enough so we can recognize them through this kind of clustering. So any of these things would be possible on novel plasmids. And in fact, Mike showed at the very end of his presentation, there are now more than 100 plasmids that seem to be circular and novel, so they are absent in all databases. There was no other way of recognizing it, but he could pull them off from complex data thanks to this clustering approach that brings together homologous genes, even though they are not identical at sequence space.
Great. Thanks, Vanshika, for the great question. And I think we're about at the hour, so I think Mike and Maren, we should thank you for the fantastic talk, and thanks for your time today. I certainly learned a lot about the microbiome.
Thank you. And I'd like to thank again to CDAC. Mike and I think this is one of the most fruitful and interesting and teaching collaborations I have going on. And people like Mike and I, although we have a lot of interest shared, very hard to come together. And we're very thankful for the support from the CDAC to kickstart this project that will hopefully end up with a nice final product very soon.
Excellent. Please go ahead, Mike.
I'll just quickly, I'll reiterate that too. I find the best science comes when you mash together very different areas, and I think that's what we've done, and I'm very excited about it. So I really appreciate the vision and what's going on with CDAC. So thank you again.
Thanks. Well, it's good. We all get to reap the rewards for these really cool talks. So thanks for educating us today. It's been fun.